Good evening, everybody. Welcome to the eighth session of uh, the course Enzyme Science and Technology in NPTEL, which is taken by Professor Vishal Trivedi from IIT Guwahati. I am Sanket, a PMRF scholar from Kusuma School of Biological Sciences, IIT Delhi. So, before we begin, we'll wait for a couple of minutes for everybody to join in, and then we will start. As usual, we will have to discuss about whatever has happened this week, and after that, we can will solve few questions which are similar to the assignments that you get. So let's see. Okay. Today's session, session 8, is about enzyme kinetics, right? So, and we'll also look at certain assays as well in this week. So, it's the eighth week out of the 12 weeks that we have. So we don't have much left. Um, if you remember, enzyme catalyzed reactions could be broadly categorized into either catabolism or anabolism. Catabolism is energy producing reactions. Anabolism is biosynthetic pathway. If we take examples, then glycolysis is, for example, produces energy. Anabolism biosynthetic pathways would be rather synthesis of a biomolecule. Okay, that would be a product. So here, energy is utilized. Here it is produced, right? So let's say examples could be like lipid biogenesis pathway or something like that. We have anyway discussed about catabolism and anabolism. So if you look at the basics, the enzyme's activity is determined by the rate of product formation or the substrate utilized in an enzyme catalyzed reaction, right? Many enzymes activity can be determined using multiple assay procedures. The choice of assay is based on the cost, availability of appropriate equipment and reagents, and the level of sensitivity that is required. It is important to ensure that any assay procedure gives the true measure of the activity of an enzyme. All the potential problems can be solved by using appropriate controls. Some seemingly abnormal behavior in assay can provide valuable insights into the property of the enzyme being studied, right? Now, if we talk about the behavior of assays, we'll talk about it. So if you see the graph here on the left, so it's the conversion percentage. So conversion is, let's say, if the substrate is forming product in the presence of an enzyme. So this is, let's say, conversion. So if we see the conversion percentage, this is how the graph looks like, right? And this is called a progress curve, right? A progress curve. And in a progress curve, we get to know what is the enzyme kinetics. So uh, the maximum, okay. So again, curve can also be represented in a graph like this. Let's draw it here. It's a V, that is the velocity, and the time can also be represented like this. So the maximum attainable V is called the V max, and half of V max is attained at okay, it is concentration, sorry, at Km. So Km is the concentration at which half of Emax is attained. That is basically, so lower the Km of an enzyme, better is its activity anyway. So enzyme can have high values of both Km and Vmax. Km and Vmax values can both be lower. Reaction rate is slowing as the enzyme is unstable and loses activity. So this is also important. The reaction rate, it slows down later on. So because substrate conversion, the initial stage is, so we'll talk about this in detail later on. Initially, the curve is linear. However, the rate of product formation begins to slow over time and linearity is lost. Now, this loss of linearity could be due to multiple reasons. One is the substrate depletion, very simple. There could be equilibrium that is attained. So what happens is, we'll see about it with the assumptions on the approaches of how we understand enzyme kinetics, where there is either we believe in equilibrium where enzyme plus substrate, they form an enzyme substrate complex which forms enzyme and product. So this equilibrium, when it is reached, then what happens is, um, when an equilibrium is reached, then the reaction slows down. So we will see, and there is then steady state formation also. We'll talk about it. Then there is product inhibition. Product inhibition is when product is produced in an due to the enzyme, when substrate is converted to product. The product sometimes, causes something called feedback inhibition. So when a lot of product is formed, it inhibits more substrate to bind to the enzyme and form more product. So product inhibition is one of the reasons why linearity in a curve is lost. Instability is another reason. Instability in any of the components of the enzyme, let's say, or on the assay. So for example, let's say if ATP is involved, if it is, the ATP is lost, then the activity is again lost. Time dependent inhibition is sometimes there. Assay method artifact could be there, and change in assay conditions like pH, temperatures, and all these kind of things will be there. So to talk about initial rate measurement is what we want to figure out. So if we have, let's say this is the graph of enzyme kinetics, this is V, and let's say concentration. This is how it looks like, right? So we want to have our reaction the kinetics at a stage like this, initial stage, right? Where there is a linear rate. So initial rate measurement, for that we can take the derivative of the early progress curve derivative or the tangent. Both are the same thing. So basically, if let's say the early tangent, early progress curve is represented by an equation called fx, function of x. So we will basically y is equal to fx. That is the basically the equation. So we will basically do dy dx. So that will be the derivative. There are other ways to extend linearity. Um, is concentration of enzyme is reduced to prolong linearity. Another, we take assumptions, is limiting the measurement of reaction. To a period where 10 to 20% of substrate is utilized, right? But it is important that entire progress curve should be monitored. Why? Because there could be like substrate depletion. Apart from substrate depletion or equilibrium, there could be other reasons, which there could be some other reasons that is causing loss in linearity. If there are other reasons behind it, which could be like, let's say, assay artifact, we have to find a way to minimize this kind of a problem. So we have to take controls and perform the experiment again. So those kind of things are there. Now there could be bursts or lags in progress curve. So what does this mean? So if we have the concentration of product on one axis and the time on time on another axis, and so the curve could be, I'll just wait. I'm trying to figure out what's happening. Okay. So curve can be like this, or the curve could be like this. Okay. So this will have different, reaction points, obviously. And if the curve is like the blue one as shown, then it is called burst, right? So there would be potential reasons behind it. So let's discuss the potential reasons, right? So the potential reasons could be inadequate temperature control. There could be settling of particles. There could be a slow detector response. Slow dissociation of a reversible inhibitor or activator, whatever. Then there could be pre-steady state transients. There could be activation by product. What does this mean? So we have talked about feedback inhibition due to product, but there could be activation as well. And there could be another reason, that is substrate interconversion. Substrate converts into some other form, that is also possible, right? So that is possible for both burst or lag. Okay, so lag is the one which is in green, right? So any of the reasons could be behind these. So initially what happens, either the reaction proceeds too fast or too slow. So that's the main goal here. Now, if we talk about the blank rate, what is a blank rate? A blank rate is the apparent rate of reaction in the absence of one of the components. And that is usually, most of the cases, it is due the substrate that is absent, but there could be other blank rates as well. So again, the potential causes for blank rate, what could be the potential causes? One is settling of the particle. Precipitation. There could be contamination. So let's say a reaction is happening even in absence of a substrate, right? In that case, particle could settling could be a reason, precipitation could be a reason, contamination could be a reason, adsorption to assay vessel. There could be some non-enzymatic reactions. Or there could be contaminating enzymes. Right? Ideally, sorry. Um, so if we see the velocity versus the enzyme concentration curve, then we will observe a curve which passes through the origin. Sorry, this is not the origin, which passes through the origin. Let's make it a little bit like this. So this is an ideal situation. Ideally, uh, increase in enzyme concentration ensures proportional increase in velocity of the reaction. Right? Now, again, there could be deviation in the linearity. Now, this deviation could be of two types. Let's look at it. We will see both the types. There could be upward curvature or there could be downward curvature. Right? In case of upward curvature, we'll see, and in case of downward curvature also, we'll see how the graph looks like. V versus the enzyme concentration, very simple for both of them. So upward curvature looks something like this. Downward curvature looks something like this. So let's find out the potential reasons behind it. So the reasons could be presence of small amount of irreversible inhibitors in the enzyme assay mixture. Another potential reason could be presence of a dissociable activator in the enzyme solution. It's very similar to the lag kind of what we observed earlier. Then there is downward curvature. Downward curvature, the reasons could be um, so detection method is limiting at high enzyme concentration. So what does this mean? So let's say if you have a lot of enzyme and a lot of product is formed, the detection limit could be achieved. So that is why we get this saturation here, right? That would be one reason. The another reason is failure to measure the initial rate of rate of reaction. So if you measure the reaction at a later stage, you will see this kind of a graph is expected. Or the third reason could be presence of dissociable inhibitor in the reaction mix. Now, how is enzyme activity expressed? So the expression is using a unit called unit itself. So usually one unit is defined as the enzyme catalyzing 1 micromolar of substrate. It could also be 1 micromole of product form based on what you are trying to detect, either the substrate depletion or product formation in 1 minute. Okay. So basically 1 unit is the micromole or of substrate used or product form per minute, right? That is the unit. If we talk about the specific activity, specific activity is the number of units per mg of the protein, of the enzyme basically, which is usually a protein. Then we have, if we know the relative molecular weight of the enzyme, then the activity can be expressed as units per micromole of enzyme. Now, according to the international, wait, just give me. According to the International Union of Biochemistry's nomenclature, the enzyme unit is 1 ketal. So 1 ketal is conversion of 1 mole substrate or the product, whatever, per second. Okay, per second. So 1 ketal, present as cat, is equal to 60 moles per minute. That is approximately 6 into 10 to the power 7 units. And 1 unit is 1 micromole per minute. That comes down to 16.67 nanoketal. So although this is the recommended unit, but it's a very high, measurement. So usually in ketal, if we do not find enzymes activity, it's always expressed in units most of the time. I'm talking about the type of enzyme assay. It could be again of multiple types. So it could be direct continuous assay. In direct continuous assay, it is the most preferred type. Difference between the substrate properties or the product, whatever, can be directly measured. So the measurement handle could be either fluorescence, pH, optical rotation, conductivity, enthalpy, viscosity, anything. It could be absorbance as well. So we'll see all of that. In the other way is indirect, right? Among indirect, there could be again 2 types, either discontinuous or continuous. Discontinuous example would also known as sampling assays. So where, let's say, if you want to find out how much ATP is produced in a reaction, so you would use a luciferase assay, luciferin. It will give you oxyluciferin plus pyrophosphates, inorganic phosphates, carbon dioxide, because it requires oxygen as well, and light, which is measured. This light is the measurement value, which is luminescence essentially. But here we cannot find the initial progress curve, cannot be actually determined because we will have the entire reaction processed. Whatever the amount of ADP is produced in the end, we will use that for sampling assay for this. In case of continuous, so it is involves performing manipulations to detect product formation. Then there could be coupled assays. So for example, uh, let's say if you want to find out hexokinase activity, which converts glucose to glucose 6-phosphate, you cannot measure this. So you have another reaction which converts glucose 6-phosphate to 6-phospho gluconolactone, which can be measured using glucose 6-phosphate dehydrogenase enzyme. So what you essentially do here is using another reaction, a coupled reaction, you're trying to find out what happens to the product. So the product is converted into another form using another reaction. So this you can measure, and this will tell you how much glucose is converted to glucose 6-phosphate. So that kind of coupled reaction can be there. Okay. Apart from this, now enzyme assay, okay, let me do it here. Enzyme assays could be photometric. That means which is the most preferred one where you would see it using light. It could be fluorescence, absorbance, anything. It could be radiometric. It could be gel electrophoresis method, or it could be chromatographic. So we'll see all of these. So photometric assay is, again, as I told you, is the most commonly used assay type. So usually light is absorbed, and we see this light absorption. It could cause change in fluorescence, absorbance, fluorescence, or the turbidity. So we'll see all of this. So if we look at light, so as you can see, the frequency changes with the spectrum. This is called an electromagnetic spectrum. So on the most left side, it would be, let's say, radio waves, then microwaves, infrared radiation. Then we have something called the visible light, which is from 400 to 750 nanometers. We have the visible light. Then we have the UV, X-ray, and the gamma rays. Fine. So the wavelength at which compound absorbs maximum amount of light, or it could be radiation, is based on the chemical properties of the compound. So there could be N to pi transition, pi to pi transitions, things like this, which contribute to this property. Absorbance is calculated A is equal to log of I by I naught, where I naught is the light transmitted, when there is no chromophore. I is the light transmitted with chromophore. A is absorbance. This is absorbance. Now, to convert the absorbance to concentration, we have the Beer-Lambert's law that tells us that absorbance is related to concentration. So this is the molar extension coefficient, which is again unique to each compound. Then we have the concentration. This is the length of cubit, which is usually 1 centimeter. So concentration can be measured as absorbance by the molar extension coefficient. So there could be certain limitations from this or this. That could be the light passing to the photodetector without, passing through the sample. So what happens is, if you have a light source here, there's a light source here, and there's a detector here, and you have the sample here. This is, let's say, the sample. If there is any light that passes, from other ends, and there are slits in between, obviously. So that kind of light has to be corrected. So absorbance in that case becomes log I plus IS by I naught plus IS, where IS is the discorrected, the light which is passing, intensity of the light that is passing without the sample. So this is the corrected absorbance. There could be some instrument errors which could also cause this problem, or noise. Turbidimetry again is used for most commonly for the concentration of, let's say, bacterial samples. We take OD measurements at 600 nanometer. So here what happens is, in your cuvette, you will have the sample which will have your bacteria as particles, and they will basically, when light falls on it, it will scatter light. So there is a process called scattering. And then this can be measured, right? It is also measured in a general spectrophotometer itself. In case of fluorescence, there is electronic transition, where what happens, your samples gets into an excited state and absorb and emits the light. So let's say this is your sample, and you give some wavelength, okay, which is called wavelength of excitation. It gets excited, gets into a higher energy state from a lower energy state, and then it comes back to a lower energy state. And in this process, it emits a light. So let's do that. So if it emits a light, that is called the emission wavelength, and we can measure this. This is called the process of fluorescence. The instrument used for this is a fluorimeter. Then there is radiometric assays as well. In this assay, what happens? We use radio label substrate, right? So usually radioisotopes are used because availability of a substrate is a problem. So like hydrogen 3H, 14C, 32 and 33P, 35S. These are all radio labeled isotopes, or 125I, which is iodine, radio labeled isotope. If they are introduced, you can measure this, and they are highly sensitive and specific. And it is used for high throughput screening. An example of radiometric assay is scintillation proximity assay, also known as SPA, where your particle will bind to a bead, which is called the SPA bead. Okay, so when the particle is bound to the bead, it will, emit beta radiation, and that will cause this bead to produce light. Now let's say if the bead and the particles are separate, it will still emit beta radiation, but this will not produce light. Okay, there will be no light. So this is how it is done. So it's appropriate for hydrolytic enzymes. Now, how to design an SPA assay? So the factors would be, let's say, the source of enzyme. Fine. There could be the design of substrate. It's common for all types of assays, actually. Signal increase or signal decrease. What are you measuring? That is, you need to know. On or off bead. Optimization of incubation conditions. Selection of the SPA bead types. Amount of the bead that needs to be optimized. Performance criteria. Like buffer and all those kind of things. And then there is validation. Fine. Now there could be assays on gel electrophoresis. So there are 5 stages to this. One is, the first one is preparation of crude enzyme extract. Then electrophoresis. Enzyme visualization. documentation, and data interpretation. So visualization means essentially the catalytic action on substrate produces a colored product. We talk about enzyme kinetics. That means basically study of enzyme reaction rate. So enzyme plus substrate forms an enzyme substrate intermediate, which forms enzyme plus product. Okay, that is how usually how it happens. And measurement of this enzyme substrate intermediate is crucial for studying enzyme kinetics. If we look at each and every symbol here, E is for enzyme, whereas if it is in the bracket, it is the concentration of enzyme. Similarly, if we write ET, it is the total enzyme, usually always in a bracket. ES is enzyme substrate complex. We already know that. K is rate constant. If we have, let's say, A, B, C, S, whatever, these are all substrates. And if we have P, Q, R, these are all product. Okay. So K, again, 1 is there, K minus 1 is there. So this is the forward reaction rate. Minus 1 is the reverse reaction rate. Kp is the catalytic rate constant. It is basically Kp. V is the velocity. V naught is the initial velocity when product is not formed. Now, there are two approaches to this. Again, as I told you, one is rapid equilibrium. Another one is steady state. So in rapid equilibrium, it is also known as Henry-Michaelis-Menten, popularly known as the Michaelis-Menten equation. Steady state is by the Briggs-Haldane. We'll study what are the assumptions in both the cases. So here we assume that the early components of a reaction are at equilibrium. So sorry. Hello. Hello. Okay. Sorry for that. Are at equilibrium. So ES, E, and S, they are all at equilibrium. Fine. In case of steady state, which is the most used approach, shortly after the reaction starts, we assume that ES would reach a steady state, which is basically close to equilibrium. Fine. So we have Henry-Michaelis-Menten equation for this, where, so how do we come at this equation? Let's see. Enzyme plus substrate gives you enzyme substrate complex, which gives you enzyme plus product, right? So ES and ES equilibrate rapidly compared to formation of enzyme and product. So the total enzyme, okay, we'll get to it, but the velocity is K2 ES and the total enzyme ET is the enzyme concentration plus the enzyme substrate concentration. So if we have these two equations and we divide the first equation with ET, so we get something like V by ET equal to K2 into ES by E plus ES, right? So this is how it looks like. Now, if you look at the reaction again, so ES can be expressed in terms of E, S, and Ks. Ks is the dissociation constant. So Ks is equal to E into S, that is equal to K minus 1. So ES substrate S by As into E. Now, if we put this is, let's say, the fourth equation. If we put the value of so okay, ES in the V by ET equation that we had previously, we will get something like this. So K2S by Ks into E by E plus S by Ks into E. So we can subtract this E from, I mean, take it out from the equation. So we will get something like V by K2 into ET S by Ks by 1 plus S by Ks. This is, let's say, equation 6. Now, if V is equal to Kp into Es. Then Kp Pt is Vmax. So this is basically Vmax. So V by Vmax is equal to S by Ks by 1 plus S by Ks. So you can write V by Vmax is equal to S. This is the derivation of the final equation Ks plus S. And this final equation is called the Michaelis-Menten equation. Fine. Okay, now we'll come to the assignments as discussed. So let's see. The enzymatic activity can be determined in enzyme catalyzed reaction by measuring the enzyme concentration, observing the change in pH, monitoring the rate of product formation or substrate consumption, or D, analyzing the reaction kinetics. So obviously, C would be the answer to this question. It is recommended to limit the initial rate measurements to a specific period of substrate consumption in order to A, observe the equilibrium phase of the reaction, allow for maximum product formation, C, maintain linearity in the reaction curve, or D, measure the enzyme concentration accurately. Actually, it is to maintain linearity. We already know. After that, linearity is lost. So if this is the velocity versus concentration curve, we get something like this, right? So the linearity is lost. Now, question 3, blank rates in enzyme assays are rates observed at the beginning of the reaction, rates observed at the end of the reaction, apparent rate observed with complete assay mixtures, apparent rate observed in the absence of one assay component. So D would be the answer. The graph typically represented when plotting initial velocity against total enzyme concentration is a curved line, is a parabolic curve, is a S-shaped curve, or a straight line passing through the origin. It's actually the straight line passing through the origin. Scenarios that can lead to an upward curvature in the relationship between initial velocity and enzyme concentration is the presence of irreversible inhibitors, lower temperature conditions, decreased ionic strength, or reduced enzyme concentration. So it would be presence of irreversible inhibitors. The main advantage of continuous indirect assays over discontinuous ones is they involve fewer manipulation. They require stopping of the reaction for measurement. They are more prone to errors. They allow determination of progress curve in a single assay. So D would be the answer. 7, the relationship between absorbance and molar absorbance coefficient. When the path length of a cuvette is 1 centimeter, is which of the following options? Is it A is equal to epsilon into C, A is equal to epsilon plus C, A is equal to epsilon by C, or A is equal to C by epsilon? So A would be epsilon into C. The A option is right. Scintillation proximity assay, or SPA, is a photometric assay, fluorometric assay, radiometric assay, or densitometric assay. It's a radiometric assay. The signal decrease scintillation proximity assay is most appropriate for hydrolases, transferases, polymerases, or kinases. So hydrolases would be the example. We already discussed it. The units of absorbance is centimeter square, meter inverse, and centimeter inverse. Molar inverse and centimeter inverse, or meter inverse, or no units. So there's no unit of absorbance. In the assay method described for the diamine oxidase, as is discussed in the lecture, the fluorophore used is a phospholipase, chitinase, elastase, or homovanillic acid. It's actually homovanillic acid. A phenomenon that contributes to nonlinearity in fluorescence-based assay at high absorbance values is photochemical reaction, inner filter effect, quenching effect, or radiation less energy transfer. So it's actually due to the inner filter effect. In the protein kinase assay, the peptide in experimental reaction migrate towards the anode because peptide has a net positive charge. or a net negative charge, or it lacks charge or has a neutral charge. So it has a negative charge. That's why it migrates towards the anode. Dehydrogenases can be visualized using a tetrazolium salt method by detecting the changes in the color of pyridine nucleotides, NADP or NADPH, observing fluorescence changes in the enzyme substrate reaction, utilizing a tetrazolium salt that becomes colored upon reduction, or visualizing the oxidation reaction without any color change. So it is using a tetrazolium salt that becomes colored. Now, the compound that is recommended as a substrate for peroxidases due to its non-carcinogenic property is N-methyl dibenzopyrazine, 3-amino ethyl carbazole, 3,3'5,5' tetramethylbenzidine, or tetrazolium salt. So it's actually 3,3'5,5' tetramethylbenzidine, also known as TMB, is used as a as a substrate for peroxidase because it is not carcinogenic. So with this, we are at the end of today's lecture. If there are any questions from your side, feel free to ask. Now, if there are no questions, then we would be calling it a session now, and we'll meet again next week at the same time, 6 to 8 p.m. So with that, I hope you had a nice session today. We'll meet again next week.